Hello everyone. Today we are going to discuss about laboratory diagnosis amoebiasis. Here there are three questions are possible to ask. One is laboratory diagnosis of amoebiasis. Other one laboratory diagnosis of intestinal amoebiasis. Third one laboratory diagnosis of extra-intestinal amoebiasis or amoebic liver abscess. According to these three questions, the answer should be varied. For example, if they ask first one, the amoebiasis laboratory diagnosis, so you have to mention both intestinal and the extra-intestinal amoebiasis. The second question, intestinal amoebiasis, you have to mention or you have to talk only the intestinal amoebiasis, how to diagnose. Third one, the extra-intestinal amoebiasis, now you have to talk only the liver abscess or the extra-intestinal amoebiasis. Before moving to the laboratory diagnosis of amoebiasis, we should know the life cycle, and if they ask any question previously, we have to give small introduction about what they ask. Here amoebiasis, we have to give small introduction. Amoebiasis is the disease caused by a entamoeba histolytica. Now we will go through the life cycle. When we consider the entamoeba histolytica, this is the amoebae, and this is only the parasite pathogenic to the human under amoebae. Other amoebas are the non-pathogenic or commercials which are not giving any pathogenic illnesses. So when we consider the life cycle, when we ingested the contaminated food and water which are containing cyst stage, which is the very resistant stages, so it is entered into our intestinal system. When we enter into the small intestine, the cyst wall is dissolved and the interior parts are come out, and when they reach into the large intestine, it is become a trophozoite, and in the large intestine they are multiplied, that means increase the number of trophozoites, and when the condition is unfavorable, it's become a cyst stage. When we shaded the stool sample of feces, the both we can see the trophozoite stage and the cyst stage. In some conditions, the trophozoite stages penetrate the colon walls of the intestine, and it is enter into the other part of the body, right? It's we call extra-intestinal amoebiasis. Especially the trophozoite stages enter into the liver, especially the main organ is the liver. We can see other organs also like lung, skin, brain and other parts also. So if it is lodged into the liver in the cells, we call extra-intestinal amoebiasis or amoebic liver abscess. In here, in extra-intestinal amoebiasis, we can only see the trophozoite stages. But in the case of other one, that means intestinal cycle, we can see both trophozoite stage and the cyst stages. Now you have some idea about the life cycle of this parasite. Initially, it contains single nucleus, and the nucleus is divided into 2 and then 4. Maximum, if it is fully matured, we can see 4 nuclei. Right? This is the specific characteristic of the entamoeba histolytica. Sometimes a mature cyst contain chromatoid bar. We can see inside the cyst of the entamoeba histolytica. Next, we will move the entamoeba coli, because entamoeba histolytica and entamoeba coli are morphologically that means the similar morphologies, some specific characteristic, but sometimes if you know only the morphological features of entamoeba histolytica and entamoeba coli, then only we can identify the pathogenic parasitical state, that means entamoeba histolytica. Sometimes we can mislead entamoeba coli as a entamoeba histolytica. So we have to differentiate these two features. Here also we can see two stages. One is trapezoid stage, other one is the cyst stage. When we consider the trapezoid stages, entamoeba coli, it is multidirectionally moving. So compare with entamoeba histolytica, this is slowly moving, and in the case of entamoeba histolytica trapezoid, it's move very faster, progressive motility. And here inside the cytoplasm of the trapezoid, here we cannot see the ingested RBC because these parasites, they don't have the ability to invade or penetrate the intestinal wall. Right, here we cannot see the ingested RBC. When we consider the cyst stages, same like entamoeba histolytica, here also trapezoid stages become a cyst stages. Initially we can see very less amount of nucleus. If it is fully matured, it become the number of nuclei become 8, right? So we have to know the morphological differences between entamoeba histolytica and the entamoeba coli, because most confusing with the entamoeba histolytica and entamoeba coli morphological forms. If you know the morphological features very well, then only you can identify the pathogenic parasites. Now we will know the differences between the entamoeba histolytica cyst and entamoeba coli cyst. This is also one of the past paper questions. So here we have to consider there are some specific characteristic of both the cyst. So we have to mention here. When we consider the size, the entamoeba coli is the larger than the entamoeba histolytica. Then in both cases, the cyst wall is double cyst wall. And when we consider the number of nuclei, in the case of entamoeba histolytica, it contains 4. If it is fully matured, we can see 4 nuclei, but in the case of entamoeba coli, we can see 8 nuclei. Then when we consider the nuclei at center, in the case of entamoeba histolytica, at center we can see in the karyosome. But in the case of entamoeba coli, karyosome present in eccentric position, that means not in center, inside the nuclei. Next step, when we consider the nuclear chromatin, there which are present surrounding the nuclear membrane, inside the nuclear membrane. In the case of entamoeba histolytica, the nuclear chromatin is evenly distributed. When we consider all the membrane area, it is the same thickness. But in the case of entamoeba coli, not like that, they unevenly distributed. When we consider the nuclear chromatin or nuclear membrane, in some places very thickness, some places no, in some places very thin nuclear chromatin. So we can use these features and we can differentiate the entamoeba coli and the entamoeba histolytica cyst. And other thing, sometimes in the case of entamoeba histolytica, we can see the chromatide bar, 1 or 2 chromatide bar in entamoeba histolytica, but we cannot see in entamoeba coli. This is the main features of the entamoeba histolytica and the entamoeba coli. Then we will move the main subject, laboratory diagnosis of intestinal amoebiasis. So if they ask laboratory diagnosis of amoebiasis, you have to mention intestinal and extra-intestinal amoebiasis. Both you have to consider. But here intestinal amoebiasis, so now you have to talk only intestinal amoebiasis, that means intestinal cycle. So mainly for the intestinal amoebiasis, we have to receive the sample is stool sample, right? So if you receive the stool sample, if it is entamoeba histolytica, the amoebiasis caused by entamoeba histolytica, first we have to look at macroscopically by using the naked eye. When we can looking the stool sample, we can get some ideas. For example, if the stool sample contain blood and mucus, we can guess that this patient is infected by entamoeba histolytica. Then we have to move to the microscopical examination. So we have to do wet mount preparation. When we are doing wet mount preparation, we have to use saline and iodine. In here, when we take the sample for the testing, we have to take the sample from where the blood and mucus is present. That area we have to take the blood sample, because that area we can see the large number of parasitical stages. Especially if we see the blood and mucus, it is acute amoebiasis. So we can see most of the time trophozoite stages. So then we can go for the concentration technique, next step. Then we can go for the permanent stain to detect the cyst and trophozoite. In the wet mount concentration technique and the permanent stain, we can detect the most of the time cyst and trophozoite. But in concentration technique, sometimes the trophozoite will ruptured, we cannot see the trophozoite stage. Then we can go for the stool culture. Here we can use Robinson culture media or diamond culture media. Then we can go for the stool antigen detection by using ELISA or ICT. Then we can go for the serological diagnosis, amoebic antigen, by using ELISA method, or we can detect the antibody by using IHA or ELISA or IFA, indirect fluorescent antibody test, or indirect hemagglutination test. Antibody detection is the indirect method. Directly we cannot identify the parasite, but we identify the antibody. By using that, we can say if the test is positive, we can say antibody, the entamoeba histolytica, is present. Then we can do for the isoenzyme analysis. Here we have to mention, in the case of intestinal amoebiasis, the entamoeba histolytica and the entamoeba dispa, these are the same species, that means same categories, and there are some differences between entamoeba histolytica and entamoeba dispa. So if we want to differentiate these two species, we have to go for the isoenzyme analysis. If the sample, if we receive the trophozoite stages, it contain the ingested RBC, we can confirm this is entamoeba histolytica, because entamoeba histolytica has a ability to invade the intestinal wall. But in the case of entamoeba dispa, they don't have that ability. That means they cannot penetrate the intestinal wall. So if by using the trophozoite stages, we can confirm first thing, if ingested RBC is present, this is confirmed entamoeba histolytica. If trophozoite does not contain the ingested RBC, that time we cannot conclude this is entamoeba histolytica, right? So here now we can guess whether it may be entamoeba histolytica or entamoeba dispa. That time we have to confirm by using isoenzyme. And same like the cyst stage, also similar morphology when we consider entamoeba histolytica and the entamoeba dispa. So if at that time we have to confirm whether it is entamoeba histolytica or entamoeba dispa, because entamoeba dispa is a common cell, but entamoeba histolytica is the pathogenic parasite. So we have to differentiate, right? Then other one is the molecular diagnosis. Here we can use PCR to detect the morphological molecular diagnosis. Then we will move laboratory diagnosis of amoebic liver abscess. So we already discussed the life cycle of the entamoeba histolytica. The entamoeba histolytica has the ability to penetrate the intestinal wall. So trophozoid stages penetrate and it goes to other part of our body. Especially they infect the liver. That's why we call amoebic liver abscess. So here we can use the biopsy sample or liver aspirate. Here we can receive the sample. So here first we will go to the microscopical examination. When we look at the microscopy, wet mount, saline and iodine, we can see the trophozoid stages only. Then we can go for the stool culture. Here why stool culture? Sometime in amoebic liver abscess, very rare amount of parasite may shaded with the stool sample. So when we culture it, we can identify by using this one also. Then antigen detection by using ELISA, and antibody detection we can do ELISA or immunofluorescence, indirect immunofluorescence or indirect heme agglutination. Antibody detection is the indirect method. By using, we can tell this patient is infected by the entamoeba histolytica, but we can confirm whether they are present or past infection. So if we want to differentiate whether it is present or past infection, we have to go for the IgG antibody and IgM antibody detection. Then other one, histopathological examination by using PS stain we can confirm. Other one, molecular diagnosis. This is most accurate one. And then we can go for the ultrasonic examination. These are the test group of tests for the amoebic liver abscess. So now we can conclude according to the question, you have to think about how to answer the question. If they ask normally amoebiasis, you have to mention intestinal amoebiasis and the liver abscess, right? But if they ask intestinal amoebiasis, you have to mention only the laboratory diagnosis of intestinal parts. If they ask amoebic liver amoebiasis, so you have to mention here in both cases specimen is vary, and other thing the laboratory diagnosis also somewhat vary. So you have to think that points. So thank you very much for your cooperation.